Assalamu alaikum, reviewer, and welcome back with this practical part number 5 and 6 of this course. The first one, genomic DNA isolation and its storage. I will give you a detailed video on that. Here you can just go through the protocols given in the slide, how we extract the DNA and which protocol, which chemicals we use for this isolation are given in these slides. Then the similarly DNA quality and quantity test. Just go through. We use the PCR. Now we can use this one. Uh, we should have a mixture for PCR. stand for master mix. Then forward and reverse primers. Then the template. Then the water. And total 10 microliter, or it could be 20, or it could be 50 microliter. But generally in my lab, I suggest my students to use 10 microliter until or unless we are going for cloning where we use the 50 microliter reaction. In regular identification of fragments, before cloning we go for 10 microliter. Then these are the conditions: initial denaturation for 5 minutes or 5 to 10 minutes from at 94. It will denature the DNA, double stranded to a single strand. Then the denaturation again for 1 minute, and this will 30 cycle. Then the annealing at 60 or any other uh, 60 degrees Celsius, 60 seconds or 45 seconds or 30 seconds depending upon your primers and your desired fragment. And then the annealing temperature, it will vary according to your primers. Then for extension 72 degrees Celsius for 60 seconds. Then final extension. This final extension is going to produce the overhang of the poly A tail, which will help us to clone it into the cloning vector such as T vectors or PUC vectors. So this PCR, it can be used for cloning, RFLP markers, AFLP markers, Dept amplification, SSTR markers, diagnostic purpose, and then finally real-time PCR is used for the gene expression profiling instead of uh, Western blotting. Thank you very much, uh, and please also watch the video related to these experiments.